Go. This is a puppy growing fast. Mitosis is the process of how cells divide and grow. So let's begin with our mitosis puppy. Starting off, we have interphase. There are two centrioles. The chromosomes are uncoiled and visible only as chromatin granules. Then we have prophase. The nucleus membrane and nucleus disappear. The replicated chromosomes form rod-shaped structures. Each pair of centrioles migrate to opposite ends of the cell, and the spindle fibers extend between the pairs of centrioles in the late process. The spindle fibers from opposite sides attach to the centromeres of their sister chromatids. Afterwards, we have metaphase, where chromosomes line up at the equator of the spindle, taking us to anaphase, where the centromeres of the sister chromatids separate. Once separated, the sister chromatids begin to move in opposite poles of the spindle. They are now called daughter chromosomes. Leading to the final stage of telophase, the new nucleus membrane forms around each new set of daughter chromosomes, creating two new nuclei. The chromosomes start to uncoil in the nuclei, while a cleavage furrow forms, which continues to constrict until the parent cell divides into two daughter cells.
This is Mother Bear. She is going through a process called meiosis inside of her. Meiosis consists of 4 stages, beginning with prophase 1. These are two sister chromatids joined at the centromere. This pairing is called synapsis. The chromosomes in the synapsis are often called tetrads because they consist of 4 chromatids. These chromosome segments then cross over with each other and exchange segments with each other. This process increases genetic variability. The chromosomes are coiled tightly to form rod-shaped structures, while the nucleus membrane and the nucleus disappear and a spindle forms. Moving to metaphase 1, the synapse chromosomes line up at the equatorial plane and attach to the spindle fibers by the centromeres. Next, we have anaphase 1, where the chromosome pairing begins to separate, while centromeres do not. This ensures that each chromosome still consists of 2 chromatids joined at the centromeres. Then the members of each chromosome pair migrate to the opposite poles of the spindle. Finally, we have telophase 1. We start to see the nucleus membrane from around each set of chromosomes as the chromosomes unwind to reform into a nucleus. Cytokinesis separates the parent cell into 2 daughter cells, with each daughter cell containing only 1 member of each chromosome pair in a replicated state, meaning each daughter cell is a haploid. So this was just meiosis 1, so please bear with me for a little more. Haha, bear. Meiosis 2 occurs after meiosis 1 with a very similar process, but in this second division of cells, the centrioles replicate again, while the chromosomes are not replicate again, since the chromosomes were just replicated. Let's briefly go through meiosis 2. Prophase 2, the nucleus membrane and the nucleus disappear and a spindle forms with the appearance of rod-shaped chromosomes. Metaphase 2, the chromosomes line up at the equator of the spindle. Each chromosome consists of 2 sister chromatids joined together at the centromere that is attached to the spindle fiber. Anaphase 2, there is a separation of the centromeres. The sister chromatids, now called daughter chromosomes, move toward opposite poles of the spindle. Telophase 2, there's as usual, the cell forms new nuclei, and cytokinesis divides the cell to form 2 haploid daughter cells. Each cell entering into meiosis 2 forms with a total of 4 haploid cells produced from the original diploid parent cell entering meiosis 1.